Good evening, everyone, and welcome to Gordon College and to these Herrmann lectures. My name is Malcolm Reed. I hung around Gordon College for about 40 years in the philosophy department, retired in 2007, and delighted to come back every year to teach in the Jerusalem-Athens Forum. But that's enough about me. Today's lecture and response marks the second of three lectures in this year's lecture series, and we would like to begin by sharing with you a little bit about the lecture's namesake, Dr. Robert Herrmann. And I'm delighted that Betty is here again. I thought she was only going to be here yesterday, here and again with her family, with Andy and Holly and others. Thank you. Please stand up so everyone can see you. Dr. Robert Herrmann is a former adjunct professor and pre-med advisor at Gordon College. He also taught medical school biochemistry for 22 years at Boston University. In 1981, Dr. Herrmann left medical education to become the executive director of the American Scientific Affiliation. That is a 2,200 member society of professionals who are interested in integrating the Christian faith and the natural sciences. As part of Dr. Herrmann's work with the American Scientific Affiliation, he met Sir John Templeton, with whom Dr. Herrmann collaborated in writing several books and also wrote a biography, a very fine one that I've read of John Templeton. We are also grateful that the John Templeton Foundation is a sponsor, a very key sponsor of this lecture series. In just a moment, I will introduce our speaker, but first, let me just give a brief overview of how we will run this time together. We will hear our Herrmann lecture speaker, and then following his lecture, we will have a response from a colleague in the field that I'll introduce later. And then finally, we'll proceed to our question and answer session, in which you will be all warmly invited to participate. And I've been asked by our respondent that you direct all your questions to the lecturer of the evening. Thank you. It's now my privilege to introduce the 2019 Herrmann Lecture Series speaker. Dr. Praveen Sethupathy is an associate professor of biomedical sciences and director of the Center for Genomics at Cornell University, where he directs a research lab focused on genomic approaches to understanding human health and diseases. He received his BA degree from Cornell University and his PhD in genomics from the University of Pennsylvania. And after completing a postdoctoral fellowship at the National Genome Research Institute under the mentorship of Dr. Francis Collins, he moved in 2011 to the University of North Carolina in Chapel Hill as an assistant professor in the Department of Genetics. The same year, he was selected by Genome Technology as one of the nation's top 25 rising young investigators in genomics. And in 2017, he returned to Cornell University as associate professor. Praveen has authored over 90 peer-reviewed publications in scientific journals and has served as a reviewer in over 35 different journal publications. Recent honors include a Faculty Merit Award for outstanding teaching and mentoring and the prestigious American Diabetes Association Pathway to Stop Diabetics Research Accelerator, which is awarded to only 3 people per year. Praveen has been an invited speaker for the Veritas Forum, has served on the advisory board for AAAS Dialogues on Science, Ethics, and Religion, and serves on the board of directors of the BioLogos Foundation, which that organization seeks to convey harmony between science and the Christian faith. He lives in Ithaca, New York, with his wife and 3 children. I haven't asked him whether they're boys, girls, or that, but maybe he'll tell us that. Okay. Thank you. Praveen, welcome to Gordon College.
Thank you. Appreciate it. Thank you very much, Malcolm. Can everybody hear me okay? Is this okay? I mentioned yesterday after the very warm introduction that the last sentence was the most important, my wife and 3 kids, so it's only fitting that I provide more information about that. I have 3 kids, a boy, girl, boy. Their ages 12, 10, and 8. And the best way to describe them is that they are my best friends. So, and I'm fond of saying that I have very little time left to teach them things. They're going to be teaching me more and more as it's becoming quickly evident. So thank you again for the welcome to Gordon College. Thank you to the Center for Faith and Inquiry for inviting me. It's really, truly an honor, particularly to do this for the Herrmann lecture series. I'm really heartened and sincerely honored that you've brought even more of your family here today. I'm hoping that means that you are keenly interested in what will come, and I hope you'll find it interesting. Yesterday, we spoke about new frontiers in biomedical research, and hopefully you found it really exciting. Hopefully some of you walked away excited about the new things that are on the horizon. But Angie, I thought, did a really good job of following up on that with ethical considerations that we ought to be carefully thinking about. And that's where we pick up today. Right? So today's lectures are going to be about some of the ethical considerations, where a Christian voice is going to be really important, and how we as a community ought to be thinking about some of these ethical challenges associated with the rapid advancement of various biotechnologies. Tomorrow, we'll have a lecture on what it means to be human, and again, hopefully you'll see some continuity of thought. Okay. So, current ethical challenges in biomedicine, there are many. I could take up this time just reading off a list of those. I don't think that would be the most productive. But I will highlight a few, talk briefly about them, and then spend most of the time on one that I have a little bit more familiarity with, and that's CRISPR-Cas9 mediated genome editing. Okay? So the first few that I'll just spend a little bit of time on. It came up in the question and answer last time. This is the privacy of human DNA databases and genetic discrimination, discriminating people on the basis of their genetic information, their DNA sequence. These are really, really important topics and questions right now, given the increased accessibility of DNA information. Right? It's only been in the last decade or a little bit more that things like Ancestry.com and 23andMe have been available. And so we haven't had to think for that long about how we store this information, how we keep it secure, how we keep it out of the hands of bad actors, and how we prevent discrimination on the basis of these. Okay? That is a large topic in and of itself, but I wanted to reissue that topic, even though I'm not going to be delving into it, so that it's on your minds and that you're aware of what's coming around the corner. There is a bill that was passed in 2008 called GINA, Genetic Information Non-Discrimination Act, and I encourage you to look it up and read about it if you're interested. It took many years for this bill to be passed, but it was really a seminal achievement. It prohibits the use of genetic information by health insurance companies and employers, right? So they can't increase your premiums on the basis of their interpretation of your DNA sequence. And employers can't take this into consideration when they're deciding whether or not to employ you for a specific job. That is a really, really, really good starting point. Right? But things like life insurance, disability insurance, long-term care insurance are not covered under that bill. Right? So it seems as though there is a grave need to actually build on GINA and provide further security for people in our communities. So the push for that is ongoing, and as I mentioned yesterday, there are actually prominent geneticists that are on the leading edge of trying to make sure this happens. Okay. Another topic. I don't know how many of you have heard about human-animal chimeras. That phrase, I'm sure, does not provide comfort, right, and is perhaps disturbing. What are these human-animal chimeras? A couple of years ago, scientists at Salk Institute created the first ever human-pig hybrid. They injected human stem cells into pig embryos, creating what's referred to as a chimera, right? A creature that consists of cells from 2 different species. Through their research, What the scientists are hoping to develop are lab-grown organs that can be used for transplantation and the saving of lives. So until I said that, I'm sure there's still some disturbed feelings, but until I said that, right, there was probably even more of a disturbed feeling. And now we're feeling, okay, well, exactly how are they doing that? How is that fitting in? But the motivation seems, at least on the surface, to be something we might be able to get on board with. Right? We want to save lives. We want to increase the number of people that are getting organ transplants. Right? Ostensibly, the motivation for this research is what the American Transplant Foundation says, and that's that every day, on average, 20 people die because there's not an available organ for transplant, and many of these individuals are kids. Right? And the more you learn about this, the more it begins to tug at your heartstrings. And the more you begin to realize that you might be open to outside-of-the-box solutions to save these kids and these people who don't have organs available to them. Again, that is not meant to justify what is happening or not justify, but it's meant to help give you a framework of how the moment we hear things, we may have a reaction. Right? But it is always a better idea to have a cooler head, learn as much as we can about it, before we offer opinions about the way that we need to proceed or what might honor or not honor God. Okay? There are obviously important safety and ethical considerations associated with this. Please don't hear from me that I'm completely on board. Okay? There's a lot of work that remains to be done, even if we do proceed with this, to minimize the risk that's associated with using organs that come out of such chimeras. There's a really important consideration of animal welfare. Right? How is all of this going to be done in a way that mitigates the pain and the suffering of animals? Right? That's an important consideration. There's also the issue of legal rights. When we start creating chimeras like this, are they human or are they animals? Humans are legal entities. Animals currently are not. So what does it mean to have a chimera? Where do they fit? Do we need a whole nother category? I'm not offering answers to these questions as much as I am saying that when we get into this kind of a business, we need to think through the implications and the ramifications. There are questions to be answered that go beyond the science. Right? It's not just about the safety and the efficacy of the procedure, it's also about the legal implications and the societal implications and things like that. Another example, brain-machine interfaces. It's another one of those terms that sort of makes us wriggle a little bit, right? Like, what does that mean? Well, let me tell you about Neuralink. A show of hands. Who knows about Neuralink? 1, 2, not that many. Elon Musk? More people know about Elon Musk. Well, Neuralink is Elon Musk. He is interested in creating a brain-machine interface, right? Sounds scary. You implant a microchip behind a person's ear, okay? And then there are tiny kind of thread-like wires that fan out into the rest of the brain or into specific parts of the brain. And they have electrodes on them, those wires, and so they can both detect brain activity and stimulate neurons, stimulate brain activity, okay? Again, the primary spoken purpose of this technology is to serve as a transformative cure for brain-related diseases like Parkinson's disease and paralysis, okay? And while it's still a little bit off, there are really some very positive indications, particularly for paralysis. There was a similar technology that was called BrainGate that Neuralink is trying to build off of that was published in 2012 by neuroscientists and engineers at Brown University that is helping paralyzed patients, people who are quadriplegic, for example, who have not been able to use their limbs for 15, 20, 25 years, using their thoughts, controlling a robotic arm to make movements, transforming and changing these people's lives. And I want to just give you an example of what this looks like. And I need Hannah's password. Thank you. All right, so you can look to your screens, whichever one is most convenient.
In this paper, two people with tetraplegia. That is, two people who are unable to move their arms or their legs in any functionally useful way.
Was that not coming up for you guys?
In this paper, two people with tetraplegia. That is, two people who are unable
It's coming? Okay.
to move their arms or their legs in any functionally useful way. We're able to control a prosthetic or a robotic arm simply by thinking about the movement of their own paralyzed hand. And they did that using the investigational BrainGate neural interface system. So they thought about using their own arm and hand as though they were reaching out themselves with their own limb, and the robotic arm moved much the way their own arm would have moved. One of the long-standing questions, not only in neuroscience, but in neurorehabilitation, is whether the cells in the motor cortex and other parts of the brain, whether they continue to function the same way years after that original injury.
It is possible for people to use their thoughts to control devices, either a computer or a robotic arm. And the way that happens is that we implant a tiny sensor, just about the size of a baby aspirin, just into the surface of the brain, and that sensor pick up the electrical impulses from a bunch of neurons. And each of those little neurons are like radio broadcast towers, putting out impulses. And when they get to the outside, a computer translator converts the pattern of pulses into something that is a command.
One of our participants was able to do something that, when all of us saw it for the first time, gave us all pause. She reached out with the robotic arm. She thought about the use of her own hand. She picked up that thermos of coffee, brought it close to her, tilted it towards herself, and sipped the coffee from a straw. And that was the first time in nearly 15 years that she had picked up anything and been able to drink from it solely of her own volition.
There was a moment of true joy, true happiness. I mean, it was beyond the fact that it was an accomplishment, I think an important advance in the entire field of brain-computer interfaces, that it was really a moment where we helped somebody do something that they had wished to do for many years. This is the culmination of more than a decade of work, beginning with science at Brown University, that laid the foundation for all of the translational work that led from monkeys playing video games using their thoughts to humans being able to control a cursor in 2006, to ultimately to today's accomplishments.
This kind of research can only happen when institutions come together and bring together teams of scientists and engineers and physicians to work closely together to create the ultimate device, one that works 24 hours a day, 7 days a week, works perfectly every time, and works for a lifetime. That's where we're all headed, and we still have some distance to get there. But to know that it's possible makes all of us quite encouraged about the future.
It gives me the goosebumps to see that. When I started and said brain-machine interfaces, I don't think many of you thought you'd be feeling the way that you're feeling right now after having watched that video. It's, again, a testimony to how we need to learn as much as we can about what the technology is, the good that it is doing, the ethical considerations surely that are present, and make holistic, provide holistic thoughts and make holistic decisions about it. Musk's technology is hoping to build on this and actually provide the feeling of touch to these robotic arms. So right now, the woman that you saw in the video, she's able to control the robotic arm with her thoughts, but is not able to feel that coffee mug as she lifts it. But what they're trying to add to this technology is that feeling of touch. Right? So really remarkable. I'm personally extraordinarily excited that there's work like this going on. Right? It represents the very best of science and technology. It seems centrally motivated by compassion for people and the desire to improve their health and their quality of life. Right? If you frame it that way, it's hard not to get behind it, right? But Musk wants to take this further. This is just the beginning for him. He wants one day to use this technology for unfiltered dissemination and augmentation, he says, of human consciousness. Right? The idea is that today, our thoughts are filtered by the imperfect means of human language and body language, right? What if we had an even more unbiased, unfiltered way to disseminate thoughts and spread it throughout the internet and have people really know what we're thinking? I don't know why we'd want that. Goodness gracious, I don't want that. But the even greater purpose behind this, he says, is that he's worried, as someone who has been a leader of artificial intelligence, he's actually a little concerned that these artificially intelligent machines are going to overtake us. So he's thinking we need to join forces, right? And we need to actually find ways in which we can merge ourselves and have some kind of a merged consciousness of AI, machines, and humans. Right? If this is sounding futuristic and sci-fi, it still is a little bit, but he's gotten far enough with these technologies that neuroscientists aren't calling him crazy, right? They're saying, we're certainly not there, right? But they see a path. They see a path to get there. Do we want to go there? That's a really, really important question. So brain-machine interfaces, I can totally get on board with that, right? But this augmentation of human consciousness things requires a little bit of attention, right? So it's the same kind of technology that can be put to use in very different ways. And it would behoove us not to throw the baby out with the bathwater and say, brain-machine interfaces all bad, right? Or human-animal chimeras may be even all bad. But actually just think about what's the good that might be able to come from it, that could be honoring to God, and what maybe isn't, right? So on that note, I want to switch to the topic that I actually want to spend more of the lecture on, and that is CRISPR-Cas9 mediated genome editing. I think a lot of people have heard the terms genome editing or CRISPR or CRISPR-Cas9. Can I have a show of hands on that? A lot of people, almost everybody, right? And I'm not surprised by that because these terms are pervasive in our society now. You hear it on NPR, you know, it's around you all the time, right? But how many of you that raised your hands said that you've heard CRISPR-Cas9 and genome editing would feel comfortable explaining what it is? Right? Craig. So, some, but the point being a small fraction of those who have heard about it, right? And those who have heard about it might even have formed opinions about it, right? But I'm not sure how much we really know or understand about it. I mean, I wouldn't unless I was in the field myself. Right? So that's what we're going to do today. We're going to talk a little bit about actually what it is. I'm not going to jump to the ethical considerations. I'm going to first just talk about what it is. Because the first few examples, I said we need to learn more, right? So I want to actually try to model that. Let's learn more together, and then we'll jump to some of the good and maybe some of the considerations. Okay. So CRISPR-Cas9, in order to understand what this is, I actually need to bring you into the world of bacteria. Okay. And I think you'll enjoy it. Give me a minute. So what CRISPR stands for is clustered, regularly interspaced, short palindromic repeats. Really wish they'd given it a shorter name, okay? And you'll hear, I've heard Nobel laureates and leaders in biomedicine talk about CRISPR on NPR and other stations, and when asked what it stands for, they can't remember off the top of their head. So if you can't for your classes, things like this, you're in good company, okay? It's clustered, regularly interspaced, short palindromic repeats. What you need to know about that, let's just break that down, okay? There are these DNA sequences in bacteria, okay? So this was actually discovered in bacterial genomes. There are these sequences that someone found that appear to repeat in the genome. Okay, so that's what the short palindromic repeats means, and these repeats happen to be palindromic, okay? And they're regularly interspaced, meaning that they're found in these repeats at regular intervals. Right? So you'll find a little sequence that is a short palindromic repeat, you'll have a little bit of filler DNA, and then you'll find another repeat, and so on. Okay, so they're at regular intervals, and they're clustered. It's not like that throughout the genome, but you'll find chunks of the genome of the bacteria that look like that. Right? So we just sort of broke that down. Does that make sense to everybody? We got clustered, regularly interspaced palindromic repeats. Okay? It was discovered, these clustered repeat regions was discovered in 1987 in Osaka University in Japan. Okay? So this is a while ago, right, that these were discovered. And then again in 1993 in Netherlands, and then in a lab in Spain. So within a span of 6 years, we had spanned a couple of continents, right, where people had discovered these repeat regions in bacterial genomes. It was not until 2007 that we actually got a clue about what they might be doing. In the intervening time, there was speculation, there were some ideas, most of them turned out to be wrong, okay? But it was not until 2007 that we actually started to figure out what these sequences were doing. And it turned out it was not so much, it's all important, but the focus shifted away from these repeats to the filler sequences between the repeats. So remember how I said there'd be a repeat, and then a little while later there'd be another repeat, and then a little while later another repeat? The filler space we just thought was filler space, so we called it spacers, okay? That shows you how important we thought it was originally, right? It turns out that's where the money is, okay? Let's talk about what that is. What is the bacteria doing there? Why does it have that? It turns out that bacteria have evolved a really clever mechanism, okay, to fight viruses that attack bacteria. Those are called bacteriophages. Okay, phage from Greek, to devour. So these bacteriophages are organisms in their own right that can attack bacteria and kill them. Okay? And there's this incredible dance of bacteriophages and bacteria in nature. And if we didn't have both, our ecosystem would be off kilter, right? Same way sort of that prey and predator are often in the same location. We need this balance. If we didn't have the bacteriophages to attack the bacteria, our world would be overrun with bacteria, right? But if we didn't have the bacteria, right, our world wouldn't be what we know of it, right? Our bodies rely on bacteria, our ecosystems, photosynthesis relies on bacteria, right? So we need bacteria, just not too much. And that's often the case in biology and in our ecosystems, right? And so there's this dance between bacteriophages and bacteria. And they keep evolving ways in which they can kind of outsmart each other, okay? The way that we have a dance with viruses that infect us, right? But bacteriophages aren't generally dangerous to us because they can't infect us. Those are called viruses that we are familiar with. These you can think of as basically viruses for bacteria specifically, and archaea and some other things, okay? So what these bacteria do, so clever, okay? When they see a virus, a bacteriophage, okay, they take a snippet of the DNA of the bacteriophage, right? So it's acquisition of that foreign material. They acquire it and they pop it into their own DNA, right? It's like a little recollection of, okay, I've seen you before, right? And if they see the virus again, what they do is they activate this thing, okay, that tells them, it's sort of a guide, it's like a self-recognition algorithm. It's a guide to the fact that it sees that same virus again, right? And that little sequence is what we call the spacer. That's what's in between these repeats. And you won't necessarily be the spacer for the same bacteriophage. You will have, that spacers will be little pieces of DNA for different bacteriophages that bacteria might see, okay? And so if you see the virus again, the bacteriophage, you use that spacer sequence, which remember was just a snippet of the DNA from the virus. You use it as a way to recognize the virus is there. And it's not traveling alone. It's traveling alongside an enzyme called Cas. And Cas just stands for CRISPR-associated, right? It's a CRISPR-associated protein. And so it's traveling alongside this guide, which we called originally the spacer. The guide then takes you to the virus, and then Cas9 is basically tethered now to the viral DNA. It's a way to bring Cas where you want it to be. What does Cas do? It cuts. So it cuts the DNA of the virus, thereby wrecking its possibility of coming inside the bacteria and replicating, right? So it's a way to kill off the bacteriophage, right? So it is fundamentally an antiviral mechanism. We have all sorts of those in our own bodies. Bacteria have them as well. And it turns out CRISPR-Cas is the bacteria's way of trying to fight against its viruses known as bacteriophages. Okay. I wanted to walk you through all of this because it's fun, but also because sometimes I get questions about why are we spending so much money in biomedicine on studying fungi and bacteria and things that don't seem so obviously related to human health and disease. Okay. I would argue that they are, but even aside from that, okay, we don't know the kinds of discoveries that can come about by studying those things, right? I gave an example earlier today in one of the classes that it was studying fungi that actually led to the discovery of statins, right, which is now one of the most widely prescribed medications in the world for hypercholesterolemia for us, right? We didn't study fungi with the idea that that would happen, right? But it came out of the studies, right? And that's the way that it was with CRISPR-Cas9. Nobody went looking for CRISPR-Cas9. They were studying bacteria, and they happened to notice something really unique about the bacterial genomes and this antiviral mechanism. Once that was popularized in the scientific community, people started to think, whoa, this is really neat. I wonder if we might be able to leverage that same thing that bacteria do to do something entirely different, edit genomes and things like that, right? So fundamentally, this just came out of wanting to understand how bacteria work, right? And I think it's really important to know that. So eventually, I mean, even though from 1987 to 2007 was the span of time that it took to understand what these were doing, from 2007 to 2013, we were, going to town on this, right? Trying to figure out how we might be able to tweak it and optimize it in order to do these kinds of gene therapy types of things. So given this, okay, why is there so much buzz about it? Right? When people started talking about leveraging CRISPR-Cas9 technology to do genome editing, why was there so much buzz? It's in part because it's a method of genetic engineering, and that those kinds of terms always get people kind of hyped up, okay? But this isn't the first time we've been doing genetic engineering, right? There have been many iterations of editing genes and editing genomes in the scientific community, right? You've heard about gene therapy cases in the past, and some success stories, some really tragic stories. But the point is that gene editing has been happening for a long time. It's been happening in the research lab for a long time. It's been attempted clinically many times before. So what was so different about this? It wasn't that we could now edit genes. I just said that we've been doing that for a long time. It was that it was the easiest, most accurate, and most cheap way of doing it than ever before. It was the democratization of gene editing, right? By and large, previous technologies were available to use by experts in an arena who had access to the best resources and the best technologies and the most money to execute genetic engineering, right? And now what we were doing with CRISPR-Cas9 was democratizing that process, right? In popular articles, you would read about how a high school student in his or her basement could do CRISPR-Cas9. I'm not quite sure we can go that far, okay? But the point was well taken, right? That this was far easier to do than ever before with any previous iteration of a genetic editing technology, right? And so what this meant was that we had to very quickly talk about all sorts of questions that are worrisome when we talk about editing genomes in a way that previously we felt there wasn't as much of an urgency or a rush, right? But if now anybody can do it with limited resources and it's very cheap and highly effective, right, at least relative to previous approaches, right, we need to really think about how we're controlling this technology, right? That's one of really the main reason why there was so much buzz around CRISPR-Cas9 technology, right? The ease with which you could do it and the accuracy with which you could edit, right? So I'm going to talk about CRISPR-Cas9 in a few different ways today. First, I'm going to talk to you about it as a research tool, where I believe there are no to little controversies, okay? Then I'm going to talk about it as a potential therapeutic approach, And I'm going to divide that into some sections where I think there probably isn't very much controversy, and in fact is really, really exciting, and some areas where maybe there's some serious, genuine controversy that we should be discussing. Okay, is that kind of a helpful outline for everybody? So as a research tool, oh my goodness, I cannot explain to you how CRISPR-Cas9 has revolutionized the work that we do in the lab. Editing genes and genomes is fundamental to the field of molecular genetics. If we have identified a gene or a rheostat that we think has an important function, the first experiment we often do is to try to knock it out or to change the sequence and see if that alters the output. And if it does, then we're able to link them together and say, oh, this gene is related to this output, and if it doesn't, then we move on to the next hypothesis, right? So molecular genetics has long been using strategies like this. But 20, 30 years ago, to manipulate a gene and be able to study then how that manipulation affects a phenotype or a trait or an output of interest might have been an entire PhD. We can do it in the lab in a week. Right? That's the remarkable transformation here, right? So we don't have to set aside all of these resources for this one gene or rheostat that we might want to study and think about the next 10 years of investigating this. We can say, well, let's try this out. No, let's try that out, right? And we can now test so many more in a span of time that is far less than what we would have needed for just testing one location of the genome before. Right? Not only that, as I alluded to previously, the accuracy with which we are able to make specific changes to the DNA, pinpoint changes, is much, much higher. Not perfect. Angie made it really clear, and it was important for her to do so, that it's an imperfect technique, and it still remains so. And we'll get to that in a minute. But it is far more effective than any other previous strategy, right? So imagine before, it took us forever to do it, and then we might end up finding that it didn't quite do it the way that we wanted. Oh boy. So much wasted time, wasted resource, right? And now we can do it much more faithfully, pinpointing exactly the location we want to change, minimizing other changes that are off target that are made, and doing so very, very quickly, right? So it's a fantastic tool. And I will give you an example from my own research of how we've leveraged it. One of the diseases that we study in my lab is a cancer called fibrolamellar carcinoma. It's a rare type of liver cancer. Okay? Until recently, we really didn't understand anything about this cancer at all. It affects children and adolescents disproportionately. And a major discovery was made about 5 years ago when it was found that there was a single mutation on one of our chromosomes that occurs that leads to, well, we didn't know at the time whether it led to the disease. We now know. But at the time, we just found that it was recurrent in patients. We just kept seeing it in patients that were coming in. Okay? And there was no other mutation that we could really see that was common among all the patients. So we thought, well, it seems like a really defensible, strong hypothesis that that mutation is somehow causing this disease. Right? I won't get into the details, but the mutation isn't just a single point mutation where one of the DNA bases kind of flips and becomes something else. It wasn't simple like that. It turned out to be a really complicated type of lesion in our DNA. Right? A type of lesion that has historically been very challenging and difficult to replicate in the lab. With CRISPR-Cas, we were able to do it in a few weeks. And it wasn't just us, it was other labs. Right? And so what we were able to do is take normal, healthy cells that we know of the liver, grow them in culture, and then execute CRISPR-Cas9 so that this particular genetic lesion at the same location and the same chromosome was made, creating that mutation, and then studying the behavior of those cells. Another lab was able to do this in mice so that we had an in vivo, a model organism that we could investigate. And in those cases, we were able to show as a field that indeed, the behavior of these cells started to change and become more cancer-like. Right? And what that suggested to us is that indeed, the presence of just this one mutation is able to initiate the tumor and contribute to its progression. And so we are now full throttle on trying to see how we can target that mutation and reverse course for these patients. Right? It's not that we would not have been able to do that with previous technology, but we just would not have been able to do it with the speed and the accuracy that we were able to do it with CRISPR-Cas, right? So that's just an example of the way that it has been revolutionizing research. But like I said, that's not where people really usually see the controversy, right? You can all kind of get on board with that, hopefully. What about CRISPR-Cas as a therapeutic intervention in live human beings? Right? It's one thing to talk about cells in a dish or even model organisms, but live human beings. And here I'm going to separate that out into two categories. What kinds of CRISPR-Cas9 editing are we doing? Are we doing it in somatic cells or are we doing it in germline cells? The distinction here is somatic cells are all the cells of your body. They're very important to you, but you're not going to pass those on, right? Or genetic changes that occur there, you're not going to pass those on, okay? Germline, sperm or egg, mutations that occur there or changes that are made there, you're going to pass on to the next generation, and your offspring will have those changes as well, okay? So that's why I'm going to draw a distinction there and talk about somatic and germline separately, okay? The somatic, as I said, focuses on a particular organ or a clump of cells in a particular location in the body. It only affects that individual, right? It has no ripple effects beyond that. And it's already being done. There are clinical trials that are popping up in our country and beyond. I'll give you a couple of examples. One, the University of Pennsylvania, right here in the States. In 2018, they commenced a trial for 2 different kinds of cancers with, I think, 6 to 8 different individuals. It was all on the level, FDA approved. They went through all of the right channels to make sure that they could do this, okay? And what they were doing was CRISPR-Cas9 to edit the specialized kinds of immune cells in these patients in order to make those immune cells less exhausted and able to fight the cancer more effectively, okay? Jury's still out. Preliminary results are promising. Really, really exciting time. Another example, sickle cell disease, okay? Sickle cell disease ravages sub-Saharan Africa, as well as many places in Central and South America. Some places in Asia and Southeast Asia, people are susceptible to sickle cell disease as well. Basically, it's prevalent where malaria is endemic. And the reason for this is because it turns out that the mutation that confers sickle cell disease also appears to provide some protection against malaria. And so it has been maintained in our population, okay? But as resources and techniques for eradicating malaria have been getting better, thanks to many organizations, not the least of which is the Gates Foundation, okay? There has been more attention placed on what we might be able to do about sickle cell. But it's not only people in sub-Saharan Africa, it's people in our country who have descent from people from sub-Saharan Africa. They're susceptible to this too, and they are not exposed to the mosquitoes that might give them malaria, right? So there have been several efforts on genetic genome editing to try to eradicate sickle cell. CRISPR-Cas9 is providing some of the greatest hope, right? So it is, the clinical trials are already ongoing. There are several examples already of individuals, not exactly with CRISPR-Cas, but with related technologies where they appear to be completely cured. And so there was a special done by 60 Minutes, I think last year, on the revolution in CRISPR-Cas and how it might eradicate sickle cell disease from the human population. And Francis Collins, the director of the NIH, showed up and dared to say that we might have a cure, right? And it's in large part because of CRISPR-Cas9 technology, right? And this hasn't really been very controversial. Why? Right? Because what we're doing is impacting a small group of cells, right, in a single individual patient, right, that isn't going to then affect anybody else in the human population. So it's not impacting the germline, it's affecting a subset of cells in an individual. That's not that unlike taking a medicine, right? When we take a medication, It's going to affect some of the cells in our body, a lot of times in ways we can't really predict. But the FDA's approved it because we think it might have more benefits than not, and so we give it a shot, right? And in many ways, this CRISPR-Cas9 is actually even more effective than that, right? There are safety considerations. For example, what if the CRISPR-Cas approach that we've used does more than the single edit that we want to do? What if it, just by chance, does something else? Those are called off-target effects, right? That remains a problem, but with every year, we're actually getting better and better and better. With every new disease that's put on the plate that we might tackle, people are finding ways to optimize CRISPR-Cas9 technology to get even better about this off-target effect. So I won't stand before you today and say that it's perfect, but I will say that the improvements have been exponential, right, in the efficacy, but also the accuracy of this technique, right? So time will tell, it remains to be seen, right? As we add more monogenic diseases, that means single mutations that lead to a disease, there are hundreds of these guys. Sickle cell is just a particularly well-known one. But there are people among us with these monogenic disorders that are suffering greatly. And there is nothing we can do to help them but CRISPR. Because we know that it's just this one mutation that's causing it in these people. There are hundreds of these kinds of cases. And so if we could have success in even some of those, like we seem to be in sickle cell, how amazing would that be, right? To be eradicating these monogenic forms of various diseases in our population. Right? Are there concerns about this? Yes, there are some safety concerns. There are, as I mentioned, about off-target effects, but they're not unlike a lot of other concerns we might have. They're not anything particularly fresh, particularly new. Okay? Where it starts to get hairy, even with somatic mutations, is if you're going after multiple mutations, right? I've been giving you examples where we know for sure that it's a single mutation that leads to the disease, right? But what if it's an accumulation of lots of different mutations that kind of increases your risk for the disease? That's getting into a gray zone, right? Because it's not as well defined. These are complex diseases that are usually governed by interactions between the unique genome and the environment that the people are in. That is not well suited to a CRISPR-Cas9 approach, right? And if we don't have that conversation and set that line as a community, then I think most will agree that we're overstepping on the utility of this technology, right? And then, of course, there's the idea of enhancement as opposed to treatment, right? But this gets usually more in the conversation about germline editing. So why don't we segue into that, okay? So there was somatic editing, but now we're going to talk about germline editing, where you actually manipulate the DNA of an embryo, okay? That embryo is going to give rise to all the cells in the body, including sperm or egg cells. And that's why germline or embryonic edits are able to be passed on to the next generation, because when that person eventually passes on to their DNA to the next generation, they're passing on their sperm or egg DNA that has those mutations or changes that were introduced when they were an embryo. That's why that's different from the somatic stuff we've been talking about, okay? Because now it affects future offspring. It's not just you making an informed decision. It's not just you going into the hospital and saying, "Doc, I get it. I know what this could potentially do to me, but I'm going to sign on the dotted line. It's okay, give it a shot," just like we might with any other drug, right? What's happening here is you're signing on the dotted line if it were to happen for generations to come. Do you really have that right? Right? Some people have argued, well, we make decisions for our kids, right? Okay, what about their kids and their kids and their kids? You're changing this in the line forever and ever, right? We don't even really have a legal framework for that, right? We don't have anything codified that even tackles that problem, right? So it's really something fundamental in our society that we need to address. Are we actually okay with that? Right? That's one of the really fundamental issues with germline CRISPR-Cas9, okay? I want to give you two examples. One is an example about hypertrophic cardiomyopathy. All you need to know about this is that it's a thickening of the heart that could then lead to sudden cardiac death. It's really quite terrible, okay? And it's about 1 in 500 adults that suffer from hypertrophic cardiomyopathy. And a lot of kids suffer from this, particularly youth that have this particular mutation and they're into sports and they will just die suddenly because of cardiac arrest. So it's really devastating, especially the disproportionate affecting of children. Oregon Health Sciences University has shown proof of principle. This hasn't been done in humans because germline editing is not allowed here in the United States. But they've shown proof of principle using cells, human cells, that they are able to reverse the mutation that is known to cause this. And when they put those cells into animal models, they have the intended effects. Right? So with this proof of principle, right, people have been asking whether we are ready to go do this in humans, right? And they did an extensive study. They showed no off-target effects on this particular one, and they'd been working on it for years. No off-target effects that they could tell, right? 100% efficacy, which is extraordinarily rare using CRISPR-Cas approaches, right? And so it's that level of achievement that got people talking as to whether maybe we should, I know this is germline and we're not supposed to do it, but gosh, this would really help these kids, right? Nothing's happened because, like I said, there's a moratorium on germline editing with CRISPR-Cas9. But it's those kinds of cases that even individuals who are wary of CRISPR-Cas9 and germline editing are saying, oh my gosh, it's that good now for this case? And we can really just reverse that mutation, right? So it tugs on the heartstrings a little bit, and that's why the conversation's really hard, okay? But any movement they might have had in that direction was totally quenched by what happened in China last year, right? And probably most of you, if not all of you, are aware, okay? Part of why I just told you a whole bunch of really good and exciting things that can happen with CRISPR is because you don't hear about those things very much. What you hear about is, this particular individual in China who they say went rogue, right? There's a bit of controversy as to how rogue he really went, because it turns out that he may have been in conversation with more people than we originally realized, more people that originally said that they were not for what he was doing. So the situation seems to be really complicated. And as for the kind of social, political kind of impact of it, I think we just have to wait and see how it all kind of shakes out. But what he did was that because China doesn't have these regulations, he went ahead in his lab and made germline edits, embryonic edits of a specific location in the genome. And 2 girls were born with genome edits. First time ever in humanity. And it was, at the time anyway, said to have been largely done without, in secret, without very much consultation or awareness from the scientific community. And it was that, in large part, that really led to a backlash from the scientific community to say, this is really, really important and controversial. And the fact that this went on without most of us being aware is really problematic and scary to people, right? What's happening out there that we don't know about, right? And even the head honchos of biomedical research in our country and beyond are not aware of these kinds of things happening in the world, doesn't inspire the greatest amount of confidence. Right? And now people have gone ahead and engineered babies, right? So this whole notion of designer babies and how everyone was trying to sort of quell that idea, oh, no, we're not talking about that. Then this happens, right? And that trust becomes mitigated, right? It's okay, well, you don't actually have as much control over this as you think you do, and it just seems like everyone's going rogue, or at least could, right? The worst part of it, if that wasn't bad enough, is that the change that was made was to a gene that would theoretically make it harder to be infected by HIV. If you were going to do it, that would have been one of the worst things to do. There's really no unmet clinical need, as has been said over and over again. We're ostensibly healthy individuals. So we're not talking about these cases that I just laid out before you. There was no prediction of hypertrophic cardiomyopathy. We weren't having sickle cell disease or any one of the other monogenic diseases, at least to our knowledge. He went in there and changed a specific location in the genome that he figured would make it harder for HIV to infect them. Okay. That was just, it was insult to injury because it wasn't even meeting an unmet clinical need, as I said, right? And so if these kinds of things can happen without too much of our knowledge, right, what else could happen? And so it has captured our imagination in all of the worst ways possible. And so all of those conversations about designer babies and going into your the doctor and saying, hey, can you do some embryonic genome editing so my kid can be taller or have bluer eyes or whatever, right, is evoking in us a period of time when eugenics was on the rise. And I'm not talking just in Europe or Germany, but here in the United States. Efforts led by scientists, right? So we have to be honest about our past in wrestling with these kinds of questions. And I've been heartened by the fact that most of the scientists in this field that I know are very sensitive to all of that, right? There are some who I think are not giving enough credence to the justifiable concern of the public. But by and large, I think there are people, especially given our history in this area, that are being extraordinarily sensitive, and they're saying this is completely unacceptable. We not only need to continue the moratorium, but we need to put together an agency, a body that will convene, hopefully an international body if we can pull it off, right? Yet another reason why having good partnerships across the world is extraordinarily important, but at the very least, a national body. We've done this before with new technologies that have come up. We convene a group of experts and we say, we bring in bioethicists and have all sorts of voices present at the table to make decisions about how we draw lines, what's going to be legal, what's not legal, and when people break the law, how do we adjudicate, right? This has not been done really yet. There have been some informal groups and conversations that have been happening, but a formal body really needs to be convened, and this is what Francis Collins and many others are hoping will happen during this time of moratorium. Right? And we want as much of the international community to join us as possible. But whether or not all of that will happen remains to be seen, but that's where we are right now. And I would contend that that's the most important next step. Before we talk anything else about germline editing of using CRISPR-Cas9, we need to keep the moratorium in place and talk it out. Because even that hasn't been done. I'm not Pollyanna about this and suggesting that once we all come to the table and talk it out, we're all gonna just hold hands and have the right set of prescriptions about this. There are some real hard conversations and problems to come. But I think we can't put the cart before the horse. Let's just at least come together and have this conversation. What I am encouraged by, and I hope you're encouraged by it as well, is that most scientists are on board with the fact that CRISPR-Cas9 is not accurate enough, not specific enough, despite the previous wonderful examples I gave you, right? And that we haven't thought enough as a society about germline editing for this to even be remotely something that we can fund or say is acceptable in the United States. Right? Vast majority of scientists I've spoken to come down that way. And so I'm encouraged by that. And there may not be as much disagreement in the community as we're afraid of, okay? But I think where the problems are going to come are in defining the line. The extreme cases are usually pretty simple. We definitely shouldn't do this. And you know what? Sickle cell disease, somatic CRISPR-Cas9, let's go for it. Let's do this as quick as we can, right? But there's a whole lot. The mess is gonna be in that gray zone where, is this a treatment or is it an enhancement? How are we thinking about this? What's going to be allowed, what's not going to be allowed? And central to that conversation is gonna be, what does it mean to be human? Right? A lot of the concern seems to come down to, are we changing our humanity? That's where the sort of playing God thing, right? It's like, DNA feels so central to who we are, so when we start mucking around with that, right? Are we changing the course of humanity in a way that is unacceptable, right? Although in reality, the medications that we've been taking for decades and decades are altering our epigenome. We just didn't know about it, so ignorance was bliss, right? So it's not as though we haven't actually wrestled with these things before. And so I think we can learn a lot from some of the conversations we've had before and not take a reactionary approach to this. To one of the student groups earlier today, one of the classes, I mentioned that, if you look back earlier in time, there were a lot of Christian communities who were against the idea of medicine, right? Because it felt unnatural. Right? It wasn't the right thing to do. If it was the will of God for my body to wither and die this way, then so be it, right? Or I should pray to such and such or to God, and if he heals me, then he heals me. If not, then no. These aren't fringe communities, right? These are communities that were dominant at some points in history, right? So I think we need to, most of us would not agree with that sentiment today. I think we'd say, oh, if we have the opportunity to heal you and cure you, I think we should go for it, right? You should take the medicines. But there was a time when that wasn't so clear to us, when we thought we were dishonoring God by doing that, right? So I think we should take as much as we can from the times in the past where we've gone through something very similar to this. It doesn't mean that we'll have the same answer. So please don't read that into what I'm saying, but it should at least inform the way that we go about having this discussion in the Christian community, right? And so if you're feeling wary about this, if you take nothing else from what I'm saying, please understand that it's rarely ever the technology that is bad or good. And that's what I'm hoping to leave you with today. It's the use of that technology, our posture toward that technology. Why are we using it? How are we using it? What is our motivation? I think that is where the rubber meets the road. That is where we're going to either be honoring God or not honoring God, right? And so I think we need to think carefully as a Christian community, because there's not only disagreement with the non-Christian community, even among Christians we're disagreeing about how to think about these technologies, right? So I think the starting point, and I know this is not an answer and I didn't intend to provide an answer today, but what I want to do is to give people enough information and knowledge and help them to see the fuzziness of this, as opposed to it being black and white, so that we can actually come to the table with a willingness to learn and with a recognition that our voice is really important. So with that, I will end today's lecture, and I hope that that was a sort of a natural segue to tomorrow's lecture on what it means to be human. Thank you very much.
Praveen, thank you so much for another splendidly crafted lecture. Thank you so much. Let me now introduce the respondent. Sharon Ketcham is Professor of Theology and Christian Ministry at Gordon College, and she earned her PhD in Theology at Boston College. Sharon's 2 decades of experience in ministry, which includes local church, researching, writing, teaching, and mentoring. And as a practical theologian, she is a scholar for the church and invites people to reflect theologically on lived Christian faith. Well, this is what many of us have been thinking about for a long time too, so this is wonderful. She's the author of Reciprocal Church, Becoming a Community Where Faith Flourishes Beyond High School, in which she proposes a new vision for a person's relationship with the church, and the accompanying values and practices that allow faith to flourish for persons and communities. Just a little note here. I spoke to a Jerusalem-Athens forum a few years ago at their inaugural meeting here at the college, and somebody asked me what did I think was the most important theological question facing their generation. And I think they were a little surprised at my answer. It was, do you understand what the church is? I think they thought that was a strange answer, but hopefully they'll think a little more about it. Sharon lives in New Hampshire with her husband and her 2 children. Sharon, welcome.
Thank you all for being here, for sitting tight for so long, a good audience. Thank you to the Herrmann family. Thank you to Hannah, Craig, and Center for Faith and Inquiry. Thank you, Dr. Praveen. You've done a good job with clear descriptions, and your enthusiasm is important to us. A student stopped me on the way to class this morning and said how thankful she was that she could track with what you were saying, a mark of a good teacher. So we're thankful to have you with us here today. You might be wondering why a theologian was invited to give a response to a scientist. I thought the same thing when I was asked. What is theology's contribution to science? Is our ethical inquiry enough? Is there a role for the lens of science? Theologian Catherine Tanner clarifies theology's role by describing it as being more than additive of a spiritual dimension. Theology's task, as we talk about it here at Gordon College, is distinct from the task of science. Theology asks questions related to God and God's relationship with the world, and this inquiry has implications for lived Christian faith. Meaning the work of theology has implications for our relationship with God, our relationship with ourselves, our relationships with one another, and our relationships with the world. So theology does not pretend to take on the work of science, and nor does science pretend to take on the work of theology. Yet these distinct disciplines, as we find them, they need one another, and they should mutually inform one another. So I accepted being here today. I was asked to do something very specific, to offer a concrete process that would enable Christians to thoughtfully engage in questions of faith and science, and to do that through specifically the lens of theology. So students who have taken Christian theology, this might become a little familiar to you per your journal prompts. Here it is. So how might we engage thoughtfully about these questions through a theological lens? I'm going to describe a three-phase process. And you know, Dr. Sethupathy today has actually spent a good deal of time on the first one, as will I. Phase 1, examine. Cool heads. We need cool heads. We need pause. We need time to think and to reflect and to wonder and to ask clear questions. To examine increases our understanding. I think there's a little bit of a precept we need to pay attention, specifically in our situation. You see, examining also helps us uncover our assumptions and our biases. Yesterday, I was thrilled to hear Dr. Sethupathy's introduction, which included what I'm going to call an apologetic of sorts, an apologetic for the positive relationship between faith and science. I might even want to call him an evangelist for this relationship. But why did we need such a defense? Why is it important that we're pausing around questions of faith and science? Do you know why? Because we have had a long history that have held them apart from one another. And that history still informs our assumptions and our biases. Where did this divide come from? And specifically, why is this divide part of maybe the generational DNA markings of Gordon College? Does that work as an analogy there? Well done. You see, I'm going to do this briefly. Christians at the beginning of the 20th century were grappling with the implications of the Enlightenment. There was increased interest and trust in the human capacity to discover and describe what was true about humanity and true about our world. And this rubbed against our interest in speaking of things related to faith. So what the church said and what Bible said. Christians were living literally amid a sociocultural epistemological shift, meaning they were wearing a certain lens and they were asked to put on a different set of glasses. I imagine what this might have looked like. A person grows up in the world where knowledge depends on the supernatural, that God reveals to us what we know and what is true, and then we encounter an Enlightenment premise that knowledge is discoverable, and especially through scientific inquiry. Guess what? Christians responded differently. I know that you're surprised by that. You might be surprised that there were really two main camps. On one side, we had what we call liberal theology. I'm not using the word liberal as a tagline for either a moral, social, or political denotation. I'm talking about the development of liberal theology, which at its core sought to accommodate the Christian faith to culture. On the other side, the fundamentalists arise, who sought to guard the Christian faith against such accommodation. Here's the key question. What is our source for determining what is true about ourselves and our world? Is it science or faith? Did you hear that? Is it this or is it that? And yep, those polarized debates, just like they do today, they broke out and these two camps emerged with an us versus them dynamic, and people would react to one another as if you were for me or against me. When we encounter polarized debates of any form, the critical space between the two sides gets lost, and we reduce complex questions to entrenched positions. This era deeply shaped our religious heritage in the United States, especially in Protestantism. There is still a for or against and this binary residue in our discussions about science and faith. So, Dr. Sethupathy serves as an apologist and an evangelist for the positive relationship for his work as a scientist and a person as faith. He still has to do that. And I'm grateful for my colleagues here at Gordon College, my scientist colleagues who do that work with our students every day. So at the very beginning, as we start to examine, what are your assumptions and your biases? How is this either or of faith and science played out in your tradition? Do you keep them separate, or do you do the hard work of trying to integrate them? How does this shape your response to scientific research and your response to theology? So we examine to uncover our assumptions and biases, but we also need to delineate pertinent questions. And I saw you doing that today and really appreciate it. What might some of those look like from a theological lens? I crafted some after yesterday's lecture and scribbled a few down today. Here they go. How does therapeutic, all from a theological lens, how does therapeutic intervention coincide or not coincide with redemption? If Christ took on the fullness of humanity and fully represents all people across race and ethnicities, how does therapeutic intervention serve all people? Might genetic engineering be a working of God's common grace to lessen the effects of sin by eliminating particular diseases, or perpetuate the effects of sin by harming disadvantaged and vulnerable people? People ask, is the research on human embryos playing God, which is a focus on God as creator? Could we expand our conversation to describe God as creator and redeemer? And what about sustainer? Does crossing the boundaries between humans with animals or machines reduce or enhance the ways we image God in the world? Or is it unrelated? If we transform our consciousness through technology, what is the role of sanctification of the will? Or is this the way to sanctify the will? I don't propose any answers. I'm just asking the questions. Do you see what I'm doing? Through a theological lens, how is it that we delineate the questions? This gives us time, first to examine our assumptions and biases, and next to begin to say, what is it that we're talking about? How does this relate to the Christian tradition? Second, so number one, examine. Number two, evaluate. How is it that we assess the questions that we ask? What is the basis of our evaluation? Theologian Janet Soskis asserts that for Christians, scientific engagement includes the preoccupation with beginnings. Our scientific engagement includes a preoccupation with beginnings. So our theological claims end up rather narrow when we only stay within a frame of creation. What if our evaluation instead followed Jürgen Moltmann's lead? This makes me excited. He claims that for far too long, eschatology acted like a bookend in theology, right? So it's in eschatology. Eschatology is what will be, the consummation of all things, God purposes as they are complete. He says, why is it that eschatology always acts like an end? So really, eschatology literally does end up being the last subject in our course in theology. Well, maybe the second to last. So guilty right here. And too often, our discussions of eschatology dissolve into speculative theories. So instead, Moltmann suggests that we flip this around and we begin our theological work with eschatology, focusing our thinking towards what will be, ultimately. So as N.T. Wright claims, we are a people who face forward towards redemption, and this orients our lives in the present, and it shapes our evaluation of the material that we've examined. So rather than engaging with science as it relates only to our beginnings, how might we evaluate biomedical research to see if it's consistent or inconsistent with redemption? How might this evaluation take place to help us focus on shalom, right-ordered relationships in societies and cultures on earth? How might this become the basis of our evaluation? Examine, evaluate, and construct. To construct is related to articulating and describing. We take seriously the responsibility as people of faith for speaking faithfully in our world, and this is hard work. We learn our lessons from history, that it's easier to dissolve into positions for and against, us versus them. Our discussions might take seriously, though, both the intent and the implications of all scientific advances. You see, constructing does involve words and also our actions. Lived Christian faith always pays attention to the implications on our relationship with God, with ourselves, with others, and with our world. You already know that there is not a one or two dimensional answer to the questions which we're facing today, that we need multi-dimensional responses. This is true for both science and theology. Maybe then all of us need to become better apologists and evangelists of science and faith dialogue, so we can work together to examine, to evaluate, and to construct responses that promote shalom. Thank you.
Thank you very much.
I had a question. The same way that we have the rights and the opportunity to choose if we want to accept and take a medicine or not, how do we take into consideration babies or even children and their rights to choose or not to go with this genome or even like just normal medicines and how that goes?
Yeah, so the question was, how do we think about the rights of children or even infants or babies with regard to genome editing, right? Again, I think it's helpful to think about what kind of editing we're doing, right? Babies, of course, don't have the capacity to make decisions for themselves as to whether or not they get a particular medication. And so parents are given the legal authority in order to make decisions on their behalf, right? So you might be able to perceive a day where that's also the case with respect to germline editing. Somatic editing, right? That could happen with sickle cell disease. It could happen with any one of these monogenic diseases that are life-threatening. There are many diseases where you know that when the baby's born, they're likely only going to have a week or two, right? And we know the mutations that cause those, right? So as long as we feel in the scientific community that the procedure is as safe as it possibly could be, and please note that we're probably not quite there for everything, and we're still working on that and getting better, But if we feel like we're in the scientific community, we're at least as safe as the medications that we tend to prescribe for infants anyway, right? And that there is consent that is properly given by the parents or whoever has legal authority, the guardians of the baby, it seems like it fits right in to what we've been doing otherwise. So that seems more of a scientific issue. Are we, happy with the efficacy and the safety of the procedure, and is the FDA going to approve it, and we'll move forward, right? The problem is really the germline editing, right? Even if we get to a point where we have a legal framework in our society where you are somehow given the right or the authority to make a decision on behalf of, a whole slew of generations after you. I'm not saying we should, okay? I'm saying even if we do, that's another layer of complication to say, can you give the right to a baby to make that decision on behalf of follow-up generations, right? So it's another layer of complication, but I would say fundamentally it's still that basic problem with germline editing of impacting the rest of your offspring, and whether or not that's the right thing to do or whether that's appropriate to do. Yeah.
Thank you. Someone else?
My question is, what implications does gene editing have on genetic diversity? You had talked about sickle cell anemia, the same mutation being positive in that it helps prevent death from malaria. Is it possible that other types of diseases could have unknown or unseen benefits to them that removing them could possibly cause a decrease in genetic diversity? Yeah.
Yes, so that is possible. There are a lot of genetic variants or mutations in the genome that, depending on the environment, right, they might have even been positively selected for because they were beneficial, provided some kind of advantage to our reproductive success, right? And so they spread in the human population. But now, because of an altered or changed environment, they're actually a liability, right? There are those kinds of examples, and then environment can change again. And maybe something is beneficial that we thought was not so. So those kinds of things definitely can happen, which is why I feel as though the black and white examples are those that are kind of early onset types of devastating diseases and disorders. I think that's a category that we can probably come together and say, irrespective of what positives they may provide that are unknown to us downstream in that person's life or someone else's, allowing them to be able to survive long enough to have a life seems like an easier answer, right? After that is when it starts to get gray, right? And there are a lot of unknowns there, and I don't think we'll ever be able to, because we can't divine the future, right? And know what a collection of variants, genetic variants, are going to offer us 200 years from now relative to what they offer us now. We just can't know, right? But I would contend that a lot of the medical choices that we make are without knowing, right? We make the best decision that we possibly can. But again, I hate to keep kind of being a broken record on this. I think where your question really becomes problematic is germline editing, right? Because again, a lot of these somatic changes that we're making are just for that individual and just for their lives, right? Even then, your question has bearing, but maybe less so.
I just had a thought as Dr. Ketcham was speaking. It's completely unthought out. I'll just say it. It strikes me that even if we were to progress at the rate we are and cure all of our known diseases through genetic engineering, I don't think we're in any danger of running out of human suffering, which kind of leads us to the path of redemption.
Hello. So my question is in regards to something that you said in class. So you commented on how one of your own personal health issues had been part of your faith transformation. And so I'm wondering if we eliminate all health issues that would contribute to faith transformation. Is that controversial or not?
Yeah.
Yeah.
Yeah, so that's a good question. I would offer, though, as a corollary to that, that I don't think that's specific to CRISPR-Cas9. I think that's a very generic question that we all could be thinking about the health profession in general, right? That we're here as a health profession to alleviate human suffering, but really to what end? Where do we, like, what's an ideal for us, right? It's related to the earlier point that is well taken, right? So I think that's a broader question. It's a good question, but a broader question. I wholeheartedly agree with you, and that was the point I was making in class, that hardship and difficulty, whether it's physical or emotional, can sometimes be a boon, right, to our growth and our relationship with others and with God, right? So I don't think the effort is necessarily to alleviate, every form of hardship, right? But I think that by and large, the focus of the health profession is to alleviate that kind of hardship that really just doesn't even allow us to have a quality of life, right? That allows us to either exist or to move on and impact other lives, right? So I think that's where the core is. You're right, there's all this gray. Even without CRISPR-Cas9, we talk about, should we be spending all this money trying to look younger as we age, right? Is that really the best use of our resources and money? And so I think even outside of CRISPR-Cas9, we should keep wrestling with those questions as a society to say, what really falls clearly under that, like, mandate of alleviating human suffering so that we can give people a chance to impact the lives of the people around them in a positive way for the glory of God, right? And being a part of the redemptive force, right? Just even if it's not so clearly a part of some grand plan that God has, or that it's a person that's then go on and be Billy Graham, I don't think we need amazing examples like that. It's just being faithful in the here and the now to say that, Christ sees a leper, he heals him, right? And so I think as images of God, we want to adopt that kind of mentality. And so in so much as is possible for me to do, I think I'll err on the side of healing. Yeah.
Take one more. Okay, yes. And this will be the last because time has gone over. Yeah.
Thank you very much for your insightful remarks. So my question touches on what you said about a technology never being good or evil, but rather it being about our use of it. And I guess it's like the age-old question about that we always ask when faced with scientific discovery. What do you think is the moral responsibility of a scientist? Should he or she continue the research and in the pursuit of truth, or should a scientist reserve things that are potentially dangerous and not publish or share that, in your opinion?
First of all, I'm glad you added he or she. That's good. Can I rephrase your question, make sure that I'm understanding it correctly? So you're saying in the line of research, if a scientist finds himself or herself in a position where they're worried about what the consequence of their finding will be or how people will interpret it, should they move ahead or not? Is that what you're saying? Yeah, I think, great question, but I think I would be I would hesitate quite a bit to give you a blanket answer, because I think part of the framework I've tried to build is that these decisions should be context dependent, right? There are, I think, precious few things in the Christian life that are just, it's like Apostles' Creed, right? But there's a lot of decisions that we make that are context dependent. We were talking in the class earlier before about how we talk about how we shouldn't lie, right? But I think we can come up with a number of circumstances where it would be more God-honoring to lie than it would be not to, right? So I think context matters a whole heck of a lot in making that decision. So I wish I could give you a blanket answer, but I think it's really going to depend on, okay, exactly what is this, right? Are other people also working on it? So if I didn't lend my voice to it, would someone else lend their voice, and it could be even worse? There are all sorts of things to consider in answering that question.
Thank you.
Thank you.